we have the epithelium, and that stroma is made up of collagen fibers, we have said. One is where we also have the canal of Schlemm, through which you is drained to the retinal nodes. For tunica we said this is the area where we have blood vessels, the choroid made up of the choroid posteriorly, the whole celiac process. I Are you getting me? Class rep, are you getting me? A class rep, are you getting me?
I'm getting.
Okay, sorry, it's a network problem here. If you can't hear, please let me know. So we've already discussed the ciliary body. I'm just reviewing what we have done, and then we'll go to the other parts. So the outer pigmented layer, which is covering the ciliary process, and also the iris, the posterior part of the iris, acts as a barrier, light and electrolyte the aqueous humor, because aqueous in electrolytes. Then we had tunic, remember? Yeah, we've done that. Stopped. Uh, 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 is originating from the transfer, then optic vesicle forms the optic cup in it, so the external and the inner part of the optic cup will differentiate only a single layer of epithelium. Inner part optic cup differentiates to form the layers of the we talked about. We have the it does not have the roads and now axons. The macula lutea is the area where we have concentration of light. It is within the optic axis, and at the center of the macula lutea is where we have the fovea centralis, and fovea centralis contain only cones. So it is an area where you have visual acuity, that is clarity of the image. The cell types in the retina, which we talked about, we have the rods and cones, which contains the photopigments. Rods contain retinine and scotopsin, that's rhodopsin, and then cones contain iodopsin, which is retinine and photopsin. So these are the ones which get the pigments are the ones which are stimulated and are able to generate an electrical impulse. This is the area of induction. And these ones here are at the outer parts of rods and cones. Then we have the horizontal cells. Horizontal cells are interneurons. They connect different parts. They connect both the rods and cones, the synapse rods and cones, and also bipolar cells. Rods and cones and bipolar cells. So they act as interneurons and therefore are able to modulate the pathway for light. Then we have the bipolar cells. Bipolar cells are connecting the rods and cones and the ganglionic cells or the optic nerve cells, optic neurons. And these bipolar cells have two processes. That's why it's called bipolar. And then amacrine cells. Amacrine cells are also acting as processes can act as an axon or a dendrite. They act as an axon or a dendrite. So they don't have very specific axon or dendrite, but depending on how the impulse is supposed to be transmitted. So amacrine cells are also important for modulating the light pathway. The other cell type that is a glial cell, specialized glial cell, which traverse the whole of the retina, is what we call the Mueller cell. The Mueller cell is a specialized glial cell. For the retina, we have this is a section of the retina. We had looked at it before. The outermost layer is pigmented layer comprising of simple cuboidal epithelia, retinal epithelia. These cells are focusing light on the outer segments of rods and cones. They also act as phagocytic. They can phagocytize. They phagocytize the extruded distal parts, outer segments of rods and cones. Outer segments of rods and cones are continuously being renewed. So these are phagocytic cells. And you can see that within the choroid, we have the blood vessels, the capillary layer, choriocapillary layer. And then from these capillaries, nutrients then pass to the retina. And because of that, this acts as a barrier towards the retina also. So they absorb material and pass the material onto the retina. Then we have these other layers here, the outer segments of rods and cones. The shape of these cells defines the name given. For the rods, the outer segments are rod-shaped. They are rod-shaped. They have a lot of lamella. The membranes are highly folded, forming a lamella, and that is to increase surface area for enzymatic activity. Enzymatic activity. Similarly, for the cones, the outer segments are cone-shaped. The cone shape. And then after the cone shape, there's a narrow part which connects it now to the body, the cell body of the rods and cones. And then from that narrow part, we then have the process that acts as an axon. Then this part here is the outer limiting membrane. And this outer limiting membrane is made up of junctional complexes between the Mueller cell, which is a glial cell, and the rods and cones. And then this part is the outer nuclear consists of cell bodies of rods and cones, cell bodies of rods and cones, but also in the inner parts here, it has cell bodies for the horizontal cells, So the processes, the dendrites for the rods and cones, which is a process, then project to this area, and also the dendrite for the horizontal cells project to this area, and synapse with the bipolar cells, the bipolar cell. So this area where the synapse is what we are referring to as the outer plexiform layer. Outer plexiform layer. And then the next layer here is the area, you can see a lot of cell bodies. So these are cell bodies for the bipolar cells. Cell bodies for the bipolar cells. So the horizontal cells, synapse at the junction between the rods and cones and also the bipolar cells, and therefore are able to modify the information that is being transmitted. And the inner nuclear layer, which is this part here, sometimes it's called granular layer because it looks like grain. The inner nuclear layer consists of cell bodies of the bipolar cells. Bipolar cells have two processes, an external process and an internal process. The inner process then project into this area where it synapses with the dendrites of the optic nerve neurons, dendrites of the optic nerve neurons, together with the dendrites of the amacrine cell, the processes of the amacrine cell. So the amacrine cell will be located in this area here and synapses with both the bipolar cells and this other layer. You can see this layer with very few neurons. This layer has very large cell bodies for the optic nerve. So we refer to this layer as the ganglionic layer. Ganglionic layer. And this ganglionic layer consists of cell bodies of the optic nerve neurons. And after that, the axons of this ganglionic layer now form this part here that we refer to as the optic nerve area. So that's now the beginning of the formation of the optic nerve. And the innermost part here is formed by junctions of the ganglionic cell and the Mueller cell. So the Mueller cell projects all the way from the outer limiting membrane to this part that we refer to as the inner limiting membrane. Because this layer here is outer and this part is inner plexiform layer, so we refer to this as the outer plexiform layer and this part as the inner plexiform layer. And this one here is the outer nuclear layer and this is the inner nuclear layer. So that is the organization of the retina. This area is the only cell types we have here are the cones. Cones. So you can see there are very few cells. So light concentrates onto this area here. This is the area we refer to as the blind spot. This is our retina here. These are the axons of the retina perforating through the sclera at the cribriform plate, perforating through the sclera. So this is the sclera, and our optic neurons perforate through now, forming the optic nerve. From there, as they continue on as the optic nerve, they are surrounded by dense fibrous equivalent to the dura mater. The dura mater, because the optic nerve is an extension of the brain. Notice here, this is the eyeball, and this is the retina. This is the chamber, the vitreous chamber, and from this vitreous chamber, the cells that we see close to the vitreous chamber are the ganglionic layer, which are cell bodies for the optic nerve. And then we have the inner plexiform layer, the inner nuclear layer, the outer plexiform layer, and the outer nuclear layer. In this part here, you can see very dark structure. This part here is the pigmented layer of the epithelium. That's what we have as the optic nerve. And then from the cornea, light passes directly to the optic nerve through the visual axis. Are you able to hear me now?
Yes.
Yes.
Thank you. I have a lot of network problems here. So this is the histology of the retina. I've said the outermost layer is the epithelial, simple cuboidal, outer segments of rods and cones, and then outer limiting membrane, outer nuclear layer, outer plexiform, inner nuclear, inner plexiform, ganglionic layer, optic nerve layer, and then inner limiting membrane. So this is what you will be able to see in our histology practical. Now, for the visual pathway, if you remember from the retina, we have the following neurons. The first order neuron are rods and cones. The second order neuron are the bipolar cells. The third order neuron are the optic nerve. So this is what we have. The optic nerve is the third order neuron after two synapses. This optic nerve from both eyes, then they cassette or they cross over at the optic chiasma. They meet at the optic chiasma. This is the optic nerve, and this is the optic chiasma. The optic chiasma is very close to the hypophysis. So if there's a tumor of the hypophysis, there'll be pressure on the optic chiasma. There'll be pressure on the optic chiasma. And then from the optic chiasma, we have the optic tract. So we call this the optic tract. This contains mixture of neurons. Now, if you notice, we have two parts of the eyeball. We have the nasal part, the one on the medial side of the retina, and we have the part lateral, that's the temporal part. So the nasal part of the retina is looking at an image that is coming from the temporal field of vision. Temporal field of vision. The temporal part of the retina is looking at an object from the nasal field of vision, close to the midline. The image is always inverted. And if we divide this into superior and inferior, the inferior part of the retina will be looking at the image from above, a superior field of vision. We need to not understand that because all the time the image is inverted. Then the inferior part of the retina is looking at an image from above, and the superior part of the retina is looking at the image from below. So that's what we have. In the case of the nasal field, nasal fibers, the axons from the nasal part of the retina, these ones cross over. They cross over at the optic chiasma. They cross to the opposite. So this part of the brain is looking at the contralateral part of the nasal, of the temporal field of vision. Contralateral is opposite. Contralateral part of the temporal field of vision because these fibers are crossing over at the optic chiasma. The fibers from the lateral part of the retina, that is the temporal part of the retina, are supposed to be looking at fibers from the medial field of vision. That is the nasal field of vision. These do not cross over. These do not cross over. So this part of the brain is looking at the ipsilateral for the same eye, ipsilateral nasal field of vision, but contralateral nasal field of vision. So this is what we have as the fibers. So these are both looking at they're looking at ipsilateral because it is on one side, is contralateral. So that is at the optic chiasma. Then we have the optic tract. The optic tract is a bundle of axons. So this terminates at the lateral geniculate body, lateral geniculate body, part of the thalamus, lateral geniculate body. The synapse at that level of the lateral geniculate body. So this is now synapsing with the fourth order neurons, which form what we refer to as the optic radiation. The optic radiation. So the optic radiation pass through the internal capsule of the cerebral cortex, projecting to the occipital lobe, the primary visual cortex in the occipital lobe. Each of the neuron carries the information from a particular spot of the field of vision, and therefore it is somatotopic. When you look at a picture, that picture is in each of these optic nerve. It is in the optic tract. It's in the lateral geniculate body, projecting to a particular place in the lateral geniculate body. It's being carried also, the same information is being carried by a neuron within the optic radiation to the occipital lobe and projects to the occipital lobe, the primary visual area, at a specific position. So there is mapping of the image throughout up to the occipital lobe. So that is the visual pathway. The optic nerve, optic chiasma, where there is crossing over of the nasal fibers, the optic tract, and then lateral geniculate body, and then optic radiation. Now, in the optic radiation, we have two pathways. We have a pathway through the temporal lobe, the lower pathway through the temporal lobe, the inferior optic radiation fibers through the temporal lobe, that we refer to as the Myers loop. So this pass through the wind around the lateral ventricle in the temporal lobe within the internal capsule, and then terminate now in the occipital lobe. Those we refer to as the Myers loop. And those ones represent the neurons from the lower quadrant, the lower half of the retina. Neurons from the lower half of the retina. And therefore, if that particular group of neurons are damaged, then you are not able to see the upper field of vision. From a horizontal field of the eye, the upper field of vision. So we refer to that as pie in the sky. A pie in the sky. There is darkness above. Pie in the sky. You can only see something that is below. So those are the fibers that are passing through the temporal lobe. The other fibers from the geniculate body pass through the parietal lobe. They pass through the parietal lobe. That's the dorsal fibers. And those ones terminate in the optic, in the visual cortex. Terminate in the visual cortex. Those fibers are the dorsal fibers originating from the dorsal fibers of the retina. So if you divide the retina into two halves, the upper half, neurons of the retina, are passing through the parietal lobe. The lower half are passing through the temporal lobe. So if the upper fibers are damaged, those particular fibers in the temporal lobe, or if the temporal lobe is affected, then you only see things that are above, but your field, the lower field, you are not able to see. So it's like pie in the floor. That's what you are calling the superior quadrantonopsia. Superior quadrantonopsia for the fibers that are passing through, damage to the fibers passing through the temporal lobe. And then fibers that pass through the parietal lobe, then you have inferior quadrantopsia, quadrantopia. This is the direct pathway for vision. Area 17, which is the primary visual cortex. From area 17, we then have some association fibers that connect to area 18 and area 19 so that you are able to recognize the object. Apart from this, we also have fibers that leave the optic tract. The same fibers from the retina that leave the septal nuclei. So those fibers are very important. And end up in the superior colliculus. They end up in the superior colliculus. We talked about superior colliculus in the midbrain. These fibers that leave the optic nerve to the superior colliculi are important in reflex activity associated with vision. The other fibers that live in the septal nuclei, those ones now are important in reflex activity that are adjusting the adjusting the pupil. Those fibers are going to trigger the Edinger-Westphal nucleus, the light reflex activity. So that is the visual pathway. I think we've talked about it. So what happens? What are the type of lesions that you have? If you have damage to the optic nerve, of course, damage to the optic nerve means the information from that eye cannot go. You can only be able to see in one eye, so that eye is blind. At the optic chiasma, if you split the optic chiasma, then you are only cutting those fibers that are a sagittal section through the optic chiasma. Median section will only cut those fibers that are originating from the nasal field of vision. Nasal field of vision. So you have blindness in the nasal field of vision, and this sometimes we see as a carpal tunnel syndrome. Fibers are looking at the temporal field of vision. Because you have cut those ones, you can only see the nasal field of vision. That means you see a very narrow mid area. Like temporal hemianopia. This one here can also occur when you have tumor with the hypophysis. When you cut the optic tract, remember, contain fibers. Each of those optic tracts contain fibers and contralateral temporal area, ipsilateral temporal retinal fibers, and contralateral eye. So that's what you will be able to see. The lateral nuclear body, if you remember, is the relay center. It's in the thalamus. So it is compressing of 6 layers. 6 layers. We have two types of neurons. Small neurons are in layer 1 and 2. Large neurons are in cell layer 1 and 2. Small neurons are in layer 3, 4, 5, and 6. Three receive ipsilateral neurons. And this layered structure is somatotopic. That means the neurons carry the same information, the same picture as you see, and because each retinal artery is. Then there are those other color, shape perception, and then fine tuning of what you are able to see. And then the interneurons, which are these coniocellular layers, act as modulators. So you can see a laterally nucleate body up. So optic radiation of the two, the Myers loop, and loop through the parietal. Sometimes when you have temporal epilepsy, they do some cutting, resection of the temporal lobe, and sometimes these fibers might be cut. Then you end up with a pie in the sky. Then you have contralateral homonymous hemianopia because they represent fibers from the opposite eye. There are other accessory visual pathways. For the papillary reflex, when you shine light into the eye, what happens? There we have the optic tract. It continues all the optic tract, then this pretectal nuclei. Remember the tectum? That is superior and inferior colliculi. We have some nuclei close to the tectum, and then it also projects to ipsilateral and contralateral Edinger-Westphal nucleus. The parasympathetic nucleus for the oculomotor. And then from the through a short ciliary and supply the sphincter papillae. Sphincter papillae. So this is how shining light into the retina will cause contraction of the pupil. Pupil contracts because the sphincter muscles are contracting. That process we call meiosis. Sometimes, if you want to see whether you see whether there's a response, brainstem death. You see whether there is a response when you shine light into the eye. Then you can define that person as being brain dead. Remember, it's going to the ipsilateral and contralateral. Because it's going to the ipsilateral and contralateral, exhibited in the opposite eye because of that crossing over to the opposite Edinger-Westphal nucleus. For accommodation, so remember for papillary reflex, it is pretectal nucleus and then Edinger-Westphal nucleus. For the accommodation, and we said accommodation is that contracting of the ciliary muscle, and that accommodation light passes all the way up to the visual cortex. All the way to the visual cortex, the visual association cortex, motor, and then through the short ciliary nerve to the ciliary muscle. So the ciliary muscle contracts shines in your eye. It's too bright, the pupil will constrict because the muscle will constrict. The sphincter papillae will contract. So that is for accommodation, but also when you are looking at a distant object, the sphincter papillae is not stimulated, but the papillae is under the influence of sympathetic nervous system. During that object that through the is applied by the ophthalmic division of the trigeminal nerve. The ophthalmic division for the trigeminal nerve for sensory supply. When you touch the cornea or an object touches the cornea, you normally have the blink reflex. And that blink reflex from the ophthalmic division, that goes to the nucleus of the trigeminal nerve, and then there's connection. The blinking reflex, because facial nerve supplies muscles of facial expression. That also acts for lacrimation. When there's irritation of the cornea, then the afference for that lacrimation is from the ophthalmic division of the trigeminal nerve, and the efference will be from the lacrimal nerve that supplies the lacrimal gland, specifically from the facial. So this just to show about the activity, consensual reflex activity. You shine light onto one eye, then the opposite eye will also act in the same manner. For gross anatomy, we have the extrinsic eye muscles, which we talked about. Muscle rectus of the middle rectus of the opposite eye is also stimulated, and the lateral rectus of the opposite eye is inhibited, so that the two eyes can change and face each other. If there's a problem with that, then you end up with strabismus. Superior recti elevate the eyeball, inferior rectus depress the eyeball, and then the oblique, I talked about the trochlea. So it comes to insert very close to the lateral rectus, and that is why So when it contracts, it will tend to pull this, so it causes depression of the eyeball. This part is depressed when the superior oblique contracts. At the same time, when you look at it from the frontal axis, when it contracts, because this is lateral, this more medial, it rotates the eyeball inwards. That is inward rotation. For the inferior oblique, the origin is more anterior compared to its insertion. Because of that, when it contracts, it will tend to elevate the eyeball, elevate this part of the eyeball, the anterior part of the eyeball. But its insertion is also more lateral compared to its origin. And therefore, when it contracts, it tends to rotate the eyeball laterally. That's what we call distortion. Superior oblique rotates medially, this one rotates laterally. So apart from the lateral rectus and superior oblique, all the muscles are supplied by the oculomotor. Lateral rectus is abducens, superior oblique is the trochlear nerve. Posterior part of the brain. That is about the visual and so let's look at audiovestibular pathway, audiovestibular apparatus. Remember, through the internal acoustic meatus is the facial nerve and vestibulocochlear nerve. Facial nerve and vestibulocochlear nerve. So vestibulocochlear nerve has two components. It has a cochlear division, which is for hearing, and it has a vestibular branch, which is for balance. That's why it's called vestibulocochlear nerve. This is the ear. So this is our auditory and also the vestibular apparatus. So our auditory and vestibular apparatus, especially for auditory, comprise of the external ear, consisting of the pinna, which is to direct the light into this external auditory canal. And then we have the tympanic membrane. So this is the external ear up to the tympanic membrane. Then we have the middle ear. This is the middle ear, and this middle ear is connected to the nasal cavity, the pharynx, through the Eustachian tube. The nasopharynx through the Eustachian tube, also referred to as pharyngotympanic tube. Pharyngotympanic. Within the middle ear, we have these bones that amplify the magnification, amplify the vibrations of the tympanic membrane. This comprises of and steps, and they have joints, and the muscles attaching to that. Some muscles are attached to the steps. We call that stapedius. The steps originates from the second pharyngeal pouch, the malleus and incus from the first pharyngeal pouch. The external acoustic meatus originates from the first pharyngeal cleft. So the outer part is lined by cutaneous. Is lined by a mucosa. Then we have the inner ear. The inner ear comprises of two parts. We have this coiled structure that we refer to as the cochlea. So the cochlea, part of the vestibular cochlear nerve, originates from the cochlea. And then we have these other parts that we refer to as semicircular canals. 3 semicircular canals. And then the middle part here, the swellings, which we refer to as the utricle and the circules, are part of the vestibular apparatus. So we are going to look at each of these to be able to define how sound is produced and how the body is balancing. The external auditory meatus is sigmoid in shape. The outer part is mainly cartilaginous, and the inner part is osseous. And the cartilaginous part is continuous with the epidermis, the skin is covering it. And they also have some hair. Of course, the hair is also part from the skin. There are sebaceous glands. Sebaceous glands. Of course, sebaceous glands are associated with the hair. And tubuloalveolar ceruminous glands, specialized sweat glands that we refer to as tubuloalveolar sweat glands. These are ceruminous glands. And these ceruminous glands produce cerumen. Cerumen is repellent to the insects. It also contains bactericidal. It also helps in trapping dust particles and so on. The inner part is very thin, is continuous to the eardrum. So the glands are missing in that part. The hair are also missing as you move towards the inner. First muscle, the first pharyngeal arch. So that tenses the tympanic membrane, and by tensing the tympanic membrane, therefore, it can influence the vibrations that take place when sound waves hit the eardrum. So structurally, the tympanic membrane externally is a cutaneous layer, it continues with the epidermis, and then internally is a mucosal layer, continuous with the lining of the nasopharynx, both cuboidal and squamous epithelia. The external part is supplied by the auricular branch of the mandibular nerve. The internal parts are supplied by the glossopharyngeal nerve, because glossopharyngeal nerve is associated with the second pharyngeal arch, which is an extension from the pharynx, from the pharyngotympanic tube, through the pharyngotympanic tube. So, because it also has a nerve from the vagus nerve, especially the lateral part of the tympanic membrane, because of that, stimulation of this part can elicit a cough reflex and even vomiting reflex. The tympanic cavity, which is the mid layer, originates from the first pharyngeal pouch, and the bones there, the malleus, the incus, and the steps, have different origin. The malleus and incus from the first pharyngeal arch, and the steps from the second pharyngeal arch. This mesenchyme from the second pharyngeal arch. Because of that, we also have a muscle that attaches to the steps. It's called stapedius muscle. So stapedius muscle is applied by the facial nerve. It's applied by the facial nerve, which is a nerve to the second pharyngeal arch. The mucosa has an epithelia, simple squamous, cuboidal, or pseudostratified. As you move towards the pharyngeal tympanic tube, it becomes pseudostratified, might be ciliated in children, and this pharyngeal tube is to balance the pressure. It's mainly to balance the pressure. Pharyngotympanic tube is to balance the pressure. And at the opening of the pharyngotympanic tube into the nasopharynx, we have adenoids. We have lymphoid tissue that form the Weldeas ring. The inner ear, we talked about it, as comprising of both the cochlea, the utricle, semicircular canals, and the saccule. So the inner ear originates from the otic placode, and this otic placode forms the otic vesicle. An otic placode then develops into an otic vesicle. The upper part of this otic vesicle differentiates to form the utricle, semicircular canals, and the endolymphatic duct. And the lower part forms the cochlea and the saccule. Cochlea and the saccule. So when you look at this placode, it's ectodermal in origin. So within the cochlea, let's look at the cochlea. Within the cochlea, we define two things. We have what we call membranous labyrinth and also bony labyrinth. These are channels. These are channels, both membranous labyrinth, within the bony labyrinth. Bony labyrinth are ducts, channel in which we have membranes. So the cochlea, from the word cochlea, it forms 2.5 turns, like a snail. It's like a snail shell. It forms 2.5 turns around a central bone that we refer to as the modiolus. 2.5 turns around the modiolus. That's what we call the cochlea. It's a spiral thing that turns 2.5 turns. And the cochlea, the 2.5 turns is the bone. Within that bony 2.5 turn channels, we have the membranous labyrinth. These membranes are usually two. We have what we call the vestibular membrane, and we have the basilar membrane, vestibular membrane, and the basilar membrane. The vestibular membrane, also called resinous membrane, so this divides the cavity into spaces. These spaces we refer to as the scala, or scali, if there are many, scala. The vestibular membrane and the basilar membrane divide the cavity of the membranous labyrinth into three subcavities. So vestibular membrane, above the vestibular membrane, we refer to that as the scala vestibuli. Below the between the vestibular and the basilar membrane, we refer to that as scala media. And then below the basilar membrane, we refer to as the scala tympani. So the scala vestibuli, scala media, and scala tympani. So these are the cavities in which we have fluid. Scala media contain what we call endolymph. It contains endolymph. Scala vestibuli and scala tympani contain perilymph. The basilar membrane is very close to the oval window, very close to the oval window, and the part close to the oval window is the proximal part, is usually stiff and narrow. Stiff and narrow. And then as you move towards the apex of the basilar membrane, it becomes thicker, so wider, and also more flexible. Because of that, the basilar membrane will be able to analyze and detect different frequencies. Close to the base, this is mainly for high frequency, detecting high frequency, and at the apex, which is wider and also more flexible, this one is mainly for low frequency. High frequency at the base, low frequency at the apex. So it's actually on a topic in terms of if you stretch the basilar membrane, you can be able to detect different frequencies at various levels. And therefore, that is important in terms of sound production. The basilar membrane also, you can divide it into what you call zona arcuata. And zona arcuata is where we have the tunnel. We're very close to the tunnel. And then zona pectinati. Zona arcuata and zona pectinati. The content of the basilar membrane is an amorphous material with some collagen fibers. So those collagen, 3 or 4 collagen fibers, they're sometimes referred to as auditory strings. Auditory strings. Because by vibration, by detecting the vibrations, they're able to produce sound. They're able to send that information, and therefore we are able to perceive the sound. Within the scala media, we have the organ of Corti, and also we have the stria vascularis. And stria vascularis is where we have the formation of the endolymph, synthesis of endolymph. This is a global structure of the cochlea. In this global structure of the cochlea, you can see repeated structures here. This is because you are taking a section, and therefore this is turning around the modulus. The 2.5 turns around the modulus. The bone, we have bone, and we also have the membranes. So bone and membranes, that's why we call membranous and also osseous labyrinth. Membranous and osseous labyrinth. We'll be able to look at a better power. So in this section of one of the channels, we have this membrane here, which is the vestibular membrane, also called resinous membrane, consisting, and this is for ionic exchange. Ionic exchange. This is the basilar membrane here. So we have these spaces, the scala vestibuli, the scala media, and scala tympani. Scala vestibuli, scala media, scala tympani. So when you look at this scala media, we have this structure that we refer to as the organ of Corti. Organ of Corti. And the organ of Corti cells rest on the basilar membrane. We have said the basilar membrane close to the oval window is stiff and narrow, detecting high frequency. And then at the apex is wider and flexible, detecting low frequency. So the organ of Corti comprises of the following. Here we have the tunnel. This tunnel here, well, divides, we have cells on this tunnel. So this divides this into two parts. These are the inner part and the outer part of the tunnel. Inner and outer part of the tunnel. We have cells that we refer to as the pillar cells. So these pillar cells are the ones which define the height of the organ of Corti, and also the boundaries between the inner and the outer. So we have inner pillar cell and outer pillar cell. Then we have sensory receptor cells here. One layer of sensory receptor cell. So we have inner hair cell, and an outer hair cell. Inner and outer hair cells. These inner and outer hair cells are supported by cells here, supporting cells. So this we refer to as phalangeal cells. Phalangeal cells, like food cells. So in the inner, we have one row of inner hair cells, one row of supporting cells. In the outer, we have 3 up to 5 rows of outer hair cells and the inner hair cells. Apart from that, we also have supporting cells. These are supporting cells, sometimes referred to as Clodia, Hansen, and Butcher. This is the stria vascularis. This is the spiral ligaments. This one here is a spiral ganglia. So let's look at the higher magnification to see the organ of Corti. Here we have tectorial membrane, and this tectorial membrane is gelatinous in nature, and the hair cells of the hair cells are embedded in the tectorial membrane. So any vibrations of the basilar membrane will cause displacement of the hair cells, the kinetocilia. That displacement of the hair cells will then lead to influx of potassium ions into the cell, and therefore generate an action potential. Tectorial membrane is produced by these denticulate cells. The inner hair cells are responsible for transduction of the mechanical movements of the membrane because of the fluid displacement, and then transmission of that to the spiral ligament here, spiral ganglia. The outer hair cells modulate the inner hair cells. So the outer hair cells are not directly responsible for the transduction of the mechanical waves to produce sound. They modulate the inner hair cells. So what happens when there's vibration of the oval window? When there's vibration of the oval window, the steps is attached to the oval window. Vibration of the oval window will cause fluid movement. That fluid movement will then cause vibration of the basilar membrane and displacement of the hair cells. Scala vestibuli and scala tympani are continuous at what we refer to as the helicotrema. Are continuous at the helicotrema. So this is what we refer to as the auditory apparatus. Auditory apparatus. So we look at the pathway from the ear. We have this spiral ligament, spiral ganglion. And then from the spiral ganglion, this then continue on as the auditory nerve to the auditory nucleus. Auditory nucleus within the pons. And then from the auditory nucleus, remember when we were talking about the brain stem, we then have the crossing over of the fibers, and that area of the crossing over is what we refer to as the trapezoid body. The fibers decassette at the trapezoid body after the cochlear nucleus. And then they ascend as the lateral lemniscus to the thalamus. Lateral lemniscus to the thalamus. And at the thalamus, at the medial geniculate body, the medial geniculate body is now the relay center with the thalamus for hearing. Then we have auditory radiation within the internal capsule to the primary auditory cortex in the temporal lobe. Primary auditory cortex in the temporal lobe. So hearing is perceived in the temporal lobe. That's where there's integration and perception within the temporal lobe. So notice the role of the inferior lateral geniculate body. Some fibers leave and enter the inferior colliculus. Those fibers that enter the inferior colliculus are very important in our auditory reflex activity. When you hear some sound, you turn your head to face the source of sound. That is because of the inferior colliculus. Remember, we talked about superior colliculus for visual, so inferior colliculus is for auditory. So this is our auditory pathway. Just to recap, the vestibular nerve enters vestibular ganglion, the nucleus within the pons, the cochlear nucleus, and then from the cochlear nucleus, fibers cross over, they decassette. After decassetting, they form the lateral lemniscus. This ascends to the medial geniculate body, and then from there as auditory radiation. Auditory radiation. Auditory radiation to the temporal lobe. Primary auditory part. Then we have those fibers that connect to the inferior colliculi, for reflex activity. So some fibers leave, enter medial inferior colliculi for auditory reflex activity. So that's what we have as the auditory pathway. Remember the trapezoid body, where some fibers decassette to the opposite. Decassette to the opposite. So the first order neuron is a spiral ganglion, and then from the cochlear nucleus, the second order neuron then synapse at the lateral geniculate body. But those for reflex activity will synapse at various levels, other levels. So the medial geniculate body, just like we saw in the lateral geniculate body, also consists of neurons in various layers. But the layers in medial geniculate body are not as organized as what we have in the lateral geniculate body. That is about hearing. So the cochlear nucleus for hearing. Now, what about balance? Vestibular cochlear, the cochlear nerve is for balance. What about balance? Again, we have bony labyrinth and membranous labyrinth. Channels within the bones, and then the membranes, which contain neuroepithelia, just like what we saw in the case of the auditory. We have the three semicircular canals, an anterior, posterior, and a lateral. Anterior, posterior, and lateral. And all these are at right angles to each other. So they define the axis, the horizontal axis, the vertical axis, the frontal axis. So that's what we have as the semicircular canals. So the movement of your head, therefore, will monitor the movement of the body and then feedback to that. Within these semicircular canals, we have the neuroepithelia forming what we call crista ambularis. Crista ambularis. Then we have the utricle and circule. Utricle and circule. And the utricle and circule also contain neuroepithelia, specialized areas of neuroepithelia that form what we call macula, the macula. For crista ambularis, the word crista means like a crest, is a raised area within semicircular canals. The membranous part of the semicircular canals, a raised area. It's made up of a raised epithelia, neuroepithelia, which contain vestibular cells. The vestibular cells contain cilia and kinetocilia. They also contain supportive cells, just like all neural tissue contain supportive cells as glial cells. And then there is a gelatinous flap, non-cellular flap, and this gelatinous flap project into the lumen of the semicircular canal at the crista ambularis. The cilia and kinetocilia of the vestibular cells are embedded within this gelatinous flap. The gelatinous flap we refer to as a cupola. Below the epithelia, we have a lamina propria, and lamina propria normally consists of loose connective tissue, and then the bony labyrinth. So you can see very elaborate, just an epithelia, very thin lamina propria, and a bony labyrinth. Displacement of the fluid within the crista umbilaris displaces the cupola, which again displaces the hair, the cilia, on the vestibular hair cells. So when that is displaced, an action potential is generated and transmitted. So crista umbilaris, by virtue of being in three planes, remember there was anterior, posterior, and lateral, all at right angles, detects angular acceleration. Angular acceleration. When you shake your head, that is angular acceleration, what we call dynamic equilibration. So when you shake your head, then you are displacing the fluid, which is displacing the cupola, which is displacing the hair cells, the cilia on the hair cells, which then generate an action potential that is detected. So you'll notice, we are young children, you turn round and round and round. And the moment you stop, you fall. Why do you fall? Because the fluid was still turning round at the time you stopped. So the brain is monitoring that you are still in motion. Try that. Turn round and round, same spot several times, and then suddenly stop. Then you fall. That is because the fluid is moving. The brain was already detecting that you are turning round. And when you stop, the fluid does not stop immediately. It still turns, displaces the cupola, displaces the hair cells, kinetocilia, and therefore you fall, you get disoriented. This is our crista umbilaris. Notice it's a crest-shaped structure. We have epithelia here, and this epithelia is neuroepithelia with hair cells. These are hair cells with hair and kinetocilia. In this area, what is missing here is our cupola, gelatinous mass. And in this area of the gelatinous mass, we have endolymph. So displacement of the fluid, displacement of the hair, and therefore an action potential is generated. And then we have the lamina propria of the loose connective tissue. So this is what we call the crista umbilaris in the semicircular canals. For the vestibular hair cells, this is the shape of the vestibular hair cells. Here they are barrel shaped. Some of them are barrel shaped, some are fusiform shaped, and it has vestibular cilia, what they call kinetocilia, and this other cilia. This is what is embedded within the cupola. These parts are embedded within the cupola. So when there's movement of the cupola, the fluid, the gelatinous structure in the cupola, then there's displacement of the kinetocilia, and an action potential is generated. That action potential then is transmitted as movement and perceived as movement. So the arterial mechanoreceptors in the neck, in crista umbilaris, and also in the macula. So displacement of the stereocilia towards kinetocilia open up potassium channels. Remember there was endolymph and very rich in potassium. They open up potassium channels, and therefore an action potential is generated. After opening up potassium channels, also calcium channels and sodium channels are opened. What about the macula? The macula is the utricle and circules. Also specialized area of the neuroepithelia. But unlike what we have in the case of the crista ambularis, this is not a crest shape. It's a more flattened shape of neuroepithelia. And within that flattened shape, we have what we call otolithic membrane. Otolithic membrane, which is a gelatinous membrane, and embedded within the gelatinous membrane, we have crystals of calcium carbonate that we refer to as autogonia. So these crystals of calcium carbonate are bearing some weight on this gelatinous membrane. Again, the neuroepithelia is vestibular cells, just like what we have seen. So any movement against gravity is acting on this. Gravity is pulling on the crystals of the autogonia, and that autogonia is putting a pressure on the otolithic membrane. That causes displacement of the kinetocilia, and subsequently potassium channels open, and then calcium channels subsequently open, and an action potential is generated. So you can see, because it's gelatinous, when you go, for example, in a lift, when you rise very fast in a lift, what happens is because the gravity is pulling on these crystals are going to cause more pressure on the otolithic membrane. When the lift stops, you find that your body was still detecting that you are in motion. That is because of that. So this one here, we have otolithic membrane, which I've talked about, the vestibular hair cells, which I've talked about. This one is mainly for linear acceleration. The linear acceleration. So gravity is acting on this linear acceleration. For the utricle, we'll be detecting horizontal acceleration, and the circules are vertical acceleration. Horizontal acceleration and vertical acceleration. You are traveling in a car, and the car suddenly stops. Your body was still moving. Your fluid was moving. Then you are able to do that. You are traveling, you are in a lift, that's vertical acceleration, and that is what is detected. So the purpose of this is to be able to give feedback to the brain, so that the brain, the cerebellum, can be able to cerebellum and the cerebral cortex can be able to regulate our muscle tone. Remember, when we are talking about cerebellum, we talked about vestibulocerebellar tract. So the vestibular apparatus, the vestibular nerve, has afferent to the cerebellum, and the cerebellum is also afferent to the vestibular apparatus. So it's a two-way monitoring system. So this is what we have as either in the utricle, the circules, the macula, in the utricle and circules. These are our hair cells, and this is our otolithic membrane. So the hair is embedded in endothelia, is embedded in the otolithic membrane. So from here, then what happens? The vestibular pathway. From the vestibular apparatus, we have vestibular ganglion, vestibular nerve, and the vestibular nerve project to the vestibular nucleus. Vestibular nucleus is in the pons. And then from the vestibular nucleus is medial longitudinal fasciculus. Remember, medial longitudinal fasciculus in the brainstem was mainly to the motor muscles involved in eye movements, so that as you move, your eyes also are detecting and getting feedback. And then we have vestibulospinal, the spinal cord For the extensor muscles, we have to the cerebellum, also mainly for balance. Vestibulospinal, remember, is for neck muscles, very important for neck muscles, and also vestibulospinal for other muscles of the body as feedback. And then vestibulocerebellar, direct to the cerebellum, but also cerebellum gives feedback the vestibular apparatus. So this is important in our regulation of balance. Regulation of balance. I'll just mention about olfactory pathway and also the gustatory. The olfactory pathway, we have already done the nasal cavity, the olfactory epithelia, and the olfactory epithelia contain Bowman's glands in which what we smell must dissolve in, and then we have olfactory cell, and from the olfactory cell we have the olfactory pathway. This is the nasal cavity. In this nasal cavity, we have the epithelia, the olfactory cells. What appears green is Bowman's glands. Bowman's glands. This is in the superior concha. In the superior concha. So from our olfactory nerve in the nasal cavity, this passes through the cribriform plate of the ethmoid bone and attaches to the synapses at the olfactory bulb. Synapses at the olfactory bulb. So within the olfactory bulb, the olfactory nerve synapses with the mitral cells and also tufted cells. The areas where the synapse is referred to as the olfactory glomerulus. Olfactory glomerulus. And then the rest, the main cells of the olfactory bulb are mitral cells and also tufted cells. Mitral cells and tufted cells. This is the structure of the olfactory bulb. It has several layers, you can see, from the outermost layer to the innermost layer. So this is fibrous outer layer, the glomerular layer, the granular layer where you have cell bodies, the plexiform layer where we have synapsing, we have the mitral cell layer, and then the inner plexiform layer and inner granular layer. All these are the layers of the olfactory bulb. Then from the olfactory bulb, if you remember what we talked when we are talking about the limbic system, is divided into two, the lateral olfactory stria and the medial olfactory stria, each of them. And then the medial olfactory stria then project to the septal nuclei. The lateral olfactory stria project the piriform cortex, that's in the temporal lobe, and also the thalamus. Piriform cortex, and the thalamus. And then the lateral project to the amygdala. And if you remember amygdala, that is mainly for emotions, part of the limbic system, responsible for emotions. And then the part that project to the septal nuclei project to the hypothalamus, again, also the limbic part of the hypothalamus. So this is how we are able to smell something. For the gustatory, the tongue, we have the taste buds. We had looked at taste buds when we are looking at the digestive system. The taste buds are within the fungiform papilla, foliate papilla, and the circumvallate papilla. Filiform papilla do not have taste buds. For the taste buds, this is taste sensation. So we have glands, serous secreting glands of von Ebner, which secrete serous fluid, in which what we taste must dissolve in. So this is circumvallate papilla with the taste buds. This is fungiform papilla. And the taste buds consist of a taste chamber a test chamber, test pore, and then within projecting into the test pore, we have the test cells, gustatory cells. And the anterior two-thirds of the tongue, we have seen, are supplied by the facial nerve, the posterior third by the glossopharyngeal nerve, and also the vagus nerve at the area of the epiglottis. This we have discussed when we are talking about the cranial nerves, both the nucleus tractus solitarius, you remember? So this is a test bud embedded among stratified squamous epithelial cells of the tongue. We had discussed this when we are looking at the tongue. So for taste perception, we have taste buds, nucleus of the tractus solitarius, remember, having a superior nucleus, the seventh cranial nerve, the facial nerve, the glossopharyngeal nerve, the posterior third of the tongue, and the vagus nerve. And then from the nucleus tractus solitarius, this passes through the medial lemniscus to posteroventral nucleus of the thalamus. And then from that posteroventral nucleus of the thalamus, we have projection to the somatosensory cortex, the area of the tongue where you have taste, that's mainly in the temporal lobe, and then the amygdala, where you have mainly associated with the limbic system, and then hippocampus. What are these parts responsible? For the amygdala and the hypothalamus, is mainly for the emotional quality of taste. Somatosensory cortex, and also part of the frontal cortex, this is conscious taste sensation. You are conscious of the taste. And then for the hippocampus, this is for the memories of taste. Memories of taste. And then this somatosensory cortex is for the perception of the taste. Whether it is bitter, sweet, sour, or umami, that perception is at the somatosensory cortex. If it is very bitter, you react. That is at the level of the amygdala. And then, for the first time, if you are testing, that is then stored as a memory. That one is by the hippocampus. So that is about our general sensation. And for the general sensation, taste and olfaction are chemical sense. Hearing is mechanical by vibration of the eardrum, which is amplified by the bones, ossicles, malleus, incus, and steps. So all that is mechanical, including our vestibular. Vision is detecting the quanta of light, And then something like touch is detecting displacement of the skin and therefore stimulating an action potential generation. So that is about our general sensory system, our sensory system. I know you have another class in 4 minutes. I'd like you to look at an assignment. But just before we look at that, I'd like to just to make a comment on this. I'd like you to look, this gross anatomy, but I'd like you to look at it from I'm saying it's gross anatomy. You will do it possibly, I don't know, you might have done it already in gross anatomy. Let me just mention it here. This is about meninges. Meninges are usually divided into pachymeninge and leptomeninges. Lepto means P and arachnoid, pachymeninge is dura mater. The functions of the meninges, these are mainly to protect the brain. That's the primary function, is protection of the brain and protection of the blood vessels. They support blood vessels. They contain and maintain cerebrospinal fluid. They form partitions within the cranial cavity. If you see the skull, the cranial cavity is very big, and the brain is floating in cerebrospinal fluid. So if there was any displacement, then the brain could very easily be damaged during that displacement. The partitions that are formed, we talked about the tendorium cerebelli, so that divides the cranial cavity into supratendorial and infratendorial compartments. Supratendorial and infratendorial compartments. Where in the infratendorial we have the brainstem and cerebellum, supratendorial we have the rest. The brain also, the meninges also support choroid plexus, but specifically the pia mater. The dura mater is divided into two. We have the meningeal part of the dura mater, and we have the periosteal part of the dura mater. The periosteal part of the dura mater forms the inner periosteum of the skull, periodium of the skull bones. Then you have the meningeal part. And the main function of this is the support of dural sinuses. And dural sinuses are spaces within the dura mater where we have blood flowing. The dural folds. If you remember, we talked about falx cerebri, falx cerebelli. This is sickle-shaped. Falx cerebri is between the two cerebral hemispheres. Falx cerebelli is between the cerebellar hemispheres. Tendorium cerebelli separates the occipital lobe from the cerebellum. Within the dural folds, we have sinuses. Sinuses are blood channels. So in falx cerebri, the superior part, we have a superior and inferior sagittal sinus. Because at the midline, that's why we call sagittal sinus. This sagittal sinus receives blood from the great cerebral vein. Falx cerebelli, associated with the falx cerebelli, we have occipital sinus, and then this receives blood from the internal vertebral venous plexus. Tendorium cerebelli on the lateral side has transverse sinus, and then at the junction where both transverse superior and inferior sagittal meet, this area is called confluence of sinuses. Confluence of sinuses. There's also petrosal sinus. And the petrosal sinus is associated with the tendorium cerebelli, most anterior. The diaphragma cell, remember, forms the roof of the hypophysis. Is pierced by the hypophyseal stalk. On either side of the diaphragma cella, we have the cavernous sinus and intercavernous sinus and petrosal sinus. These receive blood from the pterygoid plexus. And the pterygoid plexus drains the area of the danger part at the bridge of the nose. So if you are hit at the bridge of the nose, the danger triangle, this can lead into infection spreading into the petrosal sinus, cavernous sinus, and into the brain, causing meningitis. And then the sigmoid sinus joins and drains into the internal jugular vein. So these are the dural folds forming, supporting blood vessels as sinuses. So this is our falx cerebri with a superior sagittal sinus and an inferior sagittal sinus. And this lies between the two cerebral hemispheres. The two sinuses are joined by a straight sinus into this area that we refer to as the confluence of sinuses. On the lateral side of the tendorium cerebelli, we have transverse sinus here, and this transverse sinus also comes to the confluence of sinuses. And then at our area of the petrosal here, the superior petrosal, is the area draining the danger triangle of the nose, and therefore injury here can lead into injuries to this position. So this is, I'd like you to review the dural folds and also their respective sciences. Within the cavernous sinuses, most of the nerves supplying the eyeball, and also the eye muscles pass through. And therefore, if the cavernous sinus is affected, then these nerves are damaged. Sometimes you can have an aneurysm of the internal carotid, that's a bisecting aneurysm of the internal carotid. Because of that, there is pressure in the blood within the cavernous sinus, which causes pressure on the nerves, the structures passing through the cavernous sinus. And you can see an injury in the cavernous sinus of the internal carotid can lead to problems of eye muscles and also the face, innervation from the face. And abducens passes in the middle of the sinus, and therefore, if you have pressure, the first nerve that will be affected will be the abducens. The arachnoid is from the word arachnoid comes from arachnida, so it has web-like projections and is avascular. It has no blood vessels. The arachnoid has no blood vessels. It's composed of collagen, elastic, and fibroblast, and form arachnoid villi. These arachnoid villi project into the superior, especially the superior sagittal sinus, forming what you call arachnoid granulation. And the fluid, cerebrospinal fluid, drains through this villi back into the venous system. So this mainly for the drainage of cerebrospinal fluid. Below the dura, we have subdural areas, and sometimes we have injuries in these areas. And subarachnoid damages subarachnoid space. Below the arachnoid, that's what subarachnoid space. This contains CSF and also blood vessels. Because it contains CSF and blood vessels, any damage in this area, you'll be able to detect blood if you do a lumbar puncture. The CSF within the subarachnoid space, in some areas, except especially where the nerves, cranial nerves exist, where the brain is very close to the bones, they form some large CSF pool areas. Those we refer to as cisterns, specifically for protection. The largest cistern is cisterna magna at the exit of the medulla, at the exit of the spinal cord, close to the medulla oblongata. At the area of the optic chiasma, we have chiasmatic cistern. At the area of the pons, we have the pontine cistern. Between the pendangle, we have interpendangular cistern. In the midbrain, where we have the colliculi, superior and inferior colliculi, we have quadrigeminal cistern. So these ones are large pools of cerebrospinal fluid, mainly for the protection of the brain. And sometimes you have subdural bleeding of these vessels within the dura, subdura, and this can cause, you can be able to detect this in lumbar puncture, in subdural bleeding. For the pia mater, this one is fine. Pia means gentle mother. The dura means tough mother. So pia means gentle mother. And this therefore made up of fine collagen fibers, type 3 collagen fibers, the fibroblast, the elastic fibers. It also contains lymphocytes, mast cells, and is avascular. It's avascular because it's next to the blood. It gets its nutrients directly from the blood. It normally supports choroid plexus, but also support the blood vessels as they enter into the blood, into the brain, the capillaries as they enter into the brain. These dip into the sulci and fissures and follow the brain contour. Very difficult to separate it from the brain. During development, the pia mater is very important in preventing excessive migration of the neurons. If there's a problem with the pia mater, a deficit in the pia mater, then you have what is called cobblestone type of malformation. So they prevent excessive neural migration. So if you have excessive migration because of pia mater missing, then you have this cobblestone type of anomaly. So that's what we have in embryology. The choroid plexus, which is also formed by pial vessels. The choroid plexus is lined by ependyma cells, and these ependyma cells line the ventricles. They line ventricles. These are simple cuboidal epithelia with microvilli at the luminal surface, and the choroid plexus is very important for production of cerebrospinal fluid. Cerebrospinal fluid is formed in the ventricles, the lateral ventricle, the third ventricle, the fourth ventricle, and then from these ventricles, it then passes outside the ventricles through foramen of Magendie and Luschka, foramen of Magendie and Luschka, into the subarachnoid space. But it's also continuous in the central canal of the spinal cord. So that's what we have for the meninges. Please, I'd like you to go into the details of the meninges. Sometimes we have accidents When you have accidents, there's bleeding, fracture of the skull, and as a result of the bleeding, if it is extradural from the sinuses, then because the skull is a rigid structure, there's increased pressure, intracranial pressure, and that presses on the blood vessels that will push the brainstem through the foramen magnum, and that can lead to coma. That can lead to coma. In the subarachnoid, subdural spaces, you have venous sinuses. The dural layers have the venous sinuses, and those can lead also to bleeding. Remember that these dural sinuses are between the meningeal and the periosteal parts. Meningeal and periosteal parts. And also within the two layers of the dura. Subarachnoid is mainly arterial blood, and that can also lead to bleeding, and you are able to see cerebrospinal fluid. And if you have bleeding within the cerebral cortex, then you can have paralytic stroke, because that can also affect the corticospinal tract, which is mainly for motion. So I've just introduced that of the meninges. I'd like you to look at that as an exercise. This is our last class. So this last class on overview. We have the next class will be a histology practical session. One histology session. I like the class rep to organize with the technician that histology session so that you complete this particular course. You complete this particular course. Class rep, are you there?
Yes.
Thank you. Let conduct Anne so that she can be able to organize the practical session.
Okay.
Thank you. Now for the examination, the exam is going to be done on the 5th of this month. The venues will be communicated to you. I'll confirm the venues by tomorrow. So that will be communicated to you, the exam, the venues and the time. It's a very short exam, very short exam. So if you are told to be there at a specific time, you should be there even 15 minutes before the time, because if you come late, the exam would be over. So make sure you are there before time. Let everybody know that. Those who have not logged in, please, they should know that the exam is short, and therefore the venues, once confirmed, you should be at your venue to be able to start the exam on time. Thank you very much for coming to the class. When you talk about optic radiation or talk about auditory radiation, this is a big tract of neurons. Geniculocalcarine is an optic radiation because the calcarine area, the calcarine sulcus, is where we have the area 17 of the occipital lobe, the primary visual area. So it starts at the lateral geniculate body to the thalamus, which is lateral geniculate body is in the thalamus, and then the calcarine area is in the Brodmann area number 17 of the occipital lobe. Occipital lobe. The optic tract, let's say the left optic tract will be carrying fibers from the left temporal part of the retina and also the nasal part of the right eye. The nasal part of the right eye. Remember that the nasal fibers from the retina cross at the optic chiasma. They cross at the optic chiasma. Stria vascularis is an area opposite the organ of is within the opposite the organ of Corti, and stria vascularis is where we have it has a from the word stria and vascularis, we have blood vessels in that area, a strip with blood vessels, and that is where we have formation of endolymph. Formation of endolymph.